Hello students, am I audible and clearly visible? Yes. Yes. Hello students, how are you all doing? Hello, good afternoon. Yes. How are you all doing? Great. Okay. So, welcome to Unacademy NEET English channel. I am Miss Gopika, your biology educator. Okay. So, last class we were trying to understand photosynthesis in higher plants. We tried to understand chemi-osmotic hypothesis, right? So, today we are going to we also did an introduction of your C3 cycle, but today I'll explain in detail your C3 cycle and your C4 cycle, because all of you know this is very important according to your CBSE boards as well as for your NEET exams, right? Yes. So, quickly share the link with your friends so that you know this concept is really important, students. So, now you can ask me any doubts. So, quickly share the link with your friends and bring everyone in so we can have an amazing session, right? Yes. I am great. I'm great. Thank you. I'm doing great. All of you, let me know how all of you are doing. Okay, if the energy is there, if the josh is there, you know what to do in the chat box. So quickly do that and we will start our session. Okay, we will not make any delay. We will just start our session. Yes, quick. Future doctors should be more active. Yes, very good. Okay. Yes, great. Okay, students, keep your NCRT open, keep a notebook with you, a pen and a pencil, whatever stationaries that you require, because I want all of you to learn with me, okay? At the end of this class, it's going to be a very small session. At the end of this class, I want all of you to feel confident about your C3 and C4 cycle, because I have heard a lot of students telling, ma'am, I can never understand this cycle, or it is too complicated. Maybe for some of you, it might be really easy, but for some of you, it will be really complicated. So, we will break down that complex and make sure that everything enters into your brain. Okay. Yes. Hi. So many students are new to this class. Hi students, if you're new to this channel, do subscribe our channel, because we give amazing content for NEET 2024 and you can crack NEET with us. Okay. So yes. So those who are new to this class and does not know what is happening, or you know what you have to do, students, let me give you an idea. Many students have been asking us that, ma'am, can I crack NEET in 4 months? Can I crack NEET in 2 months? Or ma'am, I don't know where to start. So students, those who are confused, or those who are droppers and not really don't have an idea where to start, students, take our crash courses. We have opened crash courses where you can join, and the teachers will guide you at every step, so that in next 6 months you will be prepared to write your NEET 2024. Okay, so these are the price ranges. These are the price ranges. 1 minute. Yes, these are the price ranges. You can see we have only batches. If you just want to join a batch, it is 9,999 and 6 plus 2 months, and you get your plus and your iconic classes, where you will be put into a batch and the teachers, top educators of Unacademy will teach you. Okay, and You can look at this if you want to take iconic or if you want to take plus, depending on what you want to do at this moment. You can choose one of it, and the only thing you have to do is the description. In the description box, the link is there. Just go there and use the code GOPIKA, GOPIKA, because you're going to get amazing discounts. Okay, you're going to get really good, amazing discounts. And students, we have been telling about the UAITs, right? Quickly enroll now and write your exams and see where do you stand in your all India ranks. Okay, so these are the price ranges for that also. See, for class 12 and droppers, it is 3,999, and here for class 11, if you want to do it online, you can attempt this. Okay, so students, do not forget, always taking an exam or always attempting a mock series is very, very important, even if you're a student who does not want to study under any batch. Okay, because writing exams will boost your confidence and will also let you know where do you stand. Okay, yes. And our toolkit, I have already spoke about it. Yesterday also I told you people about it. See students, you will get all the flashcards, you will get all the mock series, you will get 15 years PYQs, everything in your hand, which are taken by our top educators, which are done by lot of assessment and testing to see if this will work. Those kind of notes are only taken and given to you. Okay, so if you want any more details about it, the link is there in the description, so just click the link and you will be able to see all of it. Okay, and only thing that you have to do is use my code. Okay, 6 letters you can remember. Right, we are studying what is this, oxalic acetic acid phosphoglycerate. You can remember GOPIKA's code, right? GOPIKA. Okay, so students, let's start our session. So understand, today's class we are going to understand what is C3 cycle and what is C4 cycle. First of all, you should understand what is the difference between this C3 cycle and C4 cycle. I have taken your NCERT lines only because you know that NCERT is what you need to crack. It's not about having additional information. So I've taken your NCERT lines, and let's mainly understand what is the difference. Okay, see, whenever we talk about say C3 cycle or C4 cycle, the number matters. What am I trying to say? I'm trying to say, if I'm talking that this is a C3 cycle, that means the first stable carbon will be a 3-carbon compound, right? It will be a 3-carbon compound. So tell me, what will be C4 cycle? If you understood this, that means if I am telling it's a C3 carbon, what does I mean? What do I mean by it? That means the carbon, the first stable component has a 3-carbon compound. Now if it is C4, tell me what is C4? Yes, very good, 4-carbon compound, right? So this will be a 4-carbon compound. This is a major difference in your C3 and C4 cycle, but there is also anatomical difference, there is also enzyme difference, all that will be different. That we will understand slowly, but as of now you understood the major difference. In C3 cycle, the first stable component is going to be a 3-carbon compound. Here the first carbon compound that you will be studying will be a 4-carbon compound. Okay, now let's see what your NCERT has to say. See, scientists also try to know whether all the plants have PGA, that is your first 3-carbon compound, okay, 3-carbon compound, as the first product of carbon dioxide fixation. Whether any other product was formed in other plants. So experiments were conducted, and then they understood that not all the plants have PGA as the first compound. They understood that there are some plants, some set of plants, okay, that has oxaloacetic acid. Okay, oxaloacetic acid, you can see OAA in your textbooks, right? OAA as your first compound. So that is when they understood, okay, then maybe there are 2 types of cycles. Okay, that is when they understood, oh, there is maybe a C3 cycle and maybe a C4 cycle, and like that. Since then, carbon dioxide assimilation during photosynthesis was said to be 2 main types. That means those plants in which the first product of carbon dioxide fixation is a C3 carbon, and the second one that was called your C3 pathway, and the second one was a C4 pathway. So only once they understood this, they came to a clarity that, oh, maybe there are 2 cycles. Is this clear to all of you? Is this clear to all of you? Can I quickly know the main difference of C4 and C3 cycle? The rest we will get into the reactions and everything, but is this clear? Quick. Students, you people are not sharing the link, you people are not liking the sessions. Okay, it's become really sad. The josh of my future doctors are in 0% now. I'm the only one who is having the josh. Is it so? Yes, so quickly like the session, quickly share the link with your friends. I want more students. I want more students here. Okay, will you all do that? You will be doing it right now. Quickly share the link with your friends. Maybe, who knows, some student might be searching, oh, how to study C3 or C4 cycle correctly. You will share a link with your friend. Your friend will thank you forever. So quickly do that. Okay, I told you people, don't do conjuicy in liking. Okay, liking the session. That is the only way you are telling me that, ma'am, I understood what you are trying to teach. Right? Yes. I am great. I am great. I am great, but the Josh is very poor, so we need to pick up that. Okay, so if this is understood, let's try to understand what is C3 cycle. Okay, students, I gave an introduction in the last class, but still I am telling for those students who joined right now. So students, C3 cycle is also called as your Calvin cycle. Okay, this is called because of the scientist who figured this out, right? Scientist who figured this out. Now, it is also called the C3 cycle because the first stable compound is a 3-carbon compound. Okay, this is the main reason. Okay, so this C3 cycle is clear. Now, what was understood? Okay, so people came up with, definitely a lot of people will come up with a lot of understanding. They thought, now carbon dioxide have to be accepted from the atmosphere. Now they thought, if one carbon dioxide has to be accepted, that means carbon dioxide has one carbon, correct? Now if one carbon, some compound I have to accept, that means the acceptor should be a 2-carbon compound. Agreed, all of you? If I have to accept 1 carbon compound, that means I should be a 2-carbon compound. Same way they thought, if it is a 1-carbon compound, then means the acceptor can be a 2-carbon compound. But that was wrong. Okay, that was wrong. How did they prove it? It was wrong. I told you they took green algae and they exposed it to sunlight and they killed it. They used some cells and they killed it with hot methanol. Like that, they tried to find the traces of carbon. Okay. Now, students, this was proven wrong. Okay, this was proven wrong. Later, they found out the first carbon dioxide acceptor is a 5-carbon compound. 5-carbon compound, which is called your RUBP. Okay, RUBP. Okay, your ribulose biphosphate. Now, this was the first compound which was capable of accepting carbon dioxide from the atmosphere. Carbon dioxide from your atmosphere. Okay. So after this finding, they put 3 main steps. Okay, they had 3 main steps for the process. That was called, first step was called your carboxylation. That means you're accepting carbon, carboxylation. And the second step was called reduction. I'll tell you what is reduction. And the third step was called regeneration. Okay, regeneration. Got it? Now, carboxylation, reduction and regeneration. These were the 3 steps. Yes, very good, Afra. So, these were the 3 steps. Okay. Now, this much is clear. This was proved wrong. That means it is not a 2-carbon compound that is going to accept carbon dioxide. It was a 5-carbon compound which was RUBP. Okay. Now, we will get into the procedure, but let me just tell you what generally, what is this, what are we talking about this, what is formed. Okay, what is formed, I'll tell you in a simple sentence. Now say, students, what formation are we talking about? Okay, now understand, you are taking carbon dioxide from the atmosphere. Okay, carbon dioxide is taking from the atmosphere, it is accepted by your RUBP. Okay, RUBP is accepting it with the help of Rubisco. Okay, and it is going to form PGA. That is your phosphoglycerate. Okay, phosphoglycerate is formed. That phosphoglycerate will be broken down. Now, here carbon dioxide was taken. Okay, RUBP is a 5-carbon compound. this 5 carbon compound is going to help to form a PGA. Okay, so let's say I'm drawing it like this. Okay, this is a 6 carbon compound. Okay, 6 carbon compound. Now I am going to, this is not a very stable compound, so they are going to break apart. Okay, so they are going to break into 3 and 3. So I am going to get G3P. That means your phosphoglyceraldehyde, or you can write your PGA, anything. Okay, phosphoglyceraldehyde. Now this phosphoglyceraldehyde will end up, okay, but here we have some people helping like NADPH. Okay, that means we are converting your PGA to an aldehyde. So NADPH with the help of ATP will do the job. Okay, will do the job and convert and give us to glucose. What am I trying to say? What am I trying to say? Okay. Now see, your carbon dioxide with the help of RUBP, with the help of Rubisco, okay, with the help of Rubisco will form PGA, will form a PGA, and that PGA will, with the help of NADPH and ATP, will form your G3P, that is your, or you can say PGAL, okay, phosphoglyceraldehyde. That aldehyde will end up giving us glucose. What are we talking about? We are talking that the carbon dioxide from outside is taken and the plants are making their own food and storing the energy. That will be, we will feed on it and we will also get energy. That's all. This is the simple process. But the complication here is because the numbers vary. Okay, numbers vary. Your Calvin cycle will at least have to undergo every time Rubisco is going to only accept 1 carbon dioxide. That means students, if I have to form glucose, how many cycles will it have to undergo? How many Calvin cycle will have to happen? Tell me quickly. Thank you, Devika. Students, tell me how many. Yes, yes, very correct. 6 cycles. All of you answer quickly. How many cycles will have to be undergone for me to form a 6 carbon glucose? Every time Rubisco is going to only accept 1, your RUBP is going to accept only 1 carbon dioxide. That means, very good, right? That means this Calvin cycle will have to go 6 times, right? 6 cycles have to be happening. This much is clear, right? Now, if this much is clear to all of you, now the steps are going to be really easy. Students, please make a note of this. Please make a note of this blue color box. This is what is going to happen. But here you are going to study with lot of number, lot of ATP and NADPH, but the procedure is the same. Okay? Yes. Okay, done? Yes. Let's see what your NCERT is trying to say. Now, carboxylation, I told you, is the first step. We try to understand that the carbon dioxide acceptor is a 5 carbon RUBP. Okay, it's a 5 carbon RUBP. Okay, I'll write here 5 carbon. So 5 carbon RUBP. Now what is going to happen? Carboxylation is the most crucial step of Calvin cycle. Why it's the most crucial step? If carbon dioxide is not coming in, then we are not going to make energy, nor the plant is going to make any food, right? Now, whereas carbon dioxide is utilized for carboxylation of RUBP, this reaction is catalyzed by an enzyme called RUBP carboxylase. That means with the, so here carbon dioxide is coming. With the help of RUBP and your Rubisco. Students, what is Rubisco's full form? Ribonuclease, rubulose biphosphate, carboxylase and oxygenase. Now, very important thing about this is, it is called as carboxylase and oxygenase. What does this mean? What does this mean? It can act as an oxygenase or it can act as a carboxylase. It has more affinity to carbon dioxide, but this can act as an oxygenase. It can act as an oxygenase when the temperature is really high. When temperature is really high or when the carbon dioxide level is very high, this Rubisco can act like an oxygenase. That means it will add the oxygen instead of accepting or adding carbon dioxide. Is this okay, students? Is this okay? in this cycle, what are we trying to form? We are trying to form glucose. That means the acceptor should be carbon dioxide. Carbon dioxide should get accepted. But if the temperature is high, or if the oxygen level is high, then this Rubisco will behave like an oxygenase. Okay, clear? This is a condition. Please make a note. It will be asked in your NEET. So make a note and keep in the textbook. High temperature and more amount of oxygen. Okay. Yes. So students, here RUBP, here Rubisco. Fine, this formation has happened. Now what are they saying? Which results in the formation of 2 molecules of your PGA. Now here this has happened. Now this conversion will form as PGA. PGA, 2 molecules of PGA. How many carbon molecules is there in PGA? How many carbon molecules is there in PGA, students? Okay, very good. How many molecules of carbon is there in your PGA? Can any of you tell me? 3? 3? Yes. Okay. So 3 PGA, that means 3 carbon. So students, here you have to understand only one thing. When I am taking carbon dioxide inside, right, for making the calculation easy, or for making the for easy understanding, I am going to take 3 molecules of carbon dioxide. That means 3 molecules of carbon dioxide is taken in by RUBP. So how many molecules of RUBP should be there to accept it? If 3 molecules of carbon dioxide has to be accepted, how many molecules of RUBP should be there? Quickly let me know. Yes, very good. 3 molecules of RUBP. I'll do one thing. I'll write all the numbers differently, different colors, so that you people will be able to calculate at the end. Okay, 3 molecule, correct, right? Now, Rubisco is that. Here we got, here how many molecules of PGA will be formed if it is 3? It will be 6 molecules, right? This is a 3 molecule, so we will get 6 molecule. Students, if it's 6 molecule, we know that one PGA has 3 carbon. That means, can I write 6? Can I write 6 into 3? One second. Yes. Yes. So, can I write? Yeah. Can I write 6 into 3? 18 carbon, 18 carbon. Yes, all of you agree? Very good, 18 carbon, correct? Now, this, what are they saying? We got 2 molecules of PGA. That means you do not have to look at the molecules here. We are taking 3 carbon. I'll tell you why we are taking 3 carbon, okay? Since this enzyme also have an oxygenation activity, it would be more correct to call it as RUBP, carboxylase oxygenase, or Rubisco, okay? Now, this PGA will get converted to, 1,3-diphosphoglycerate. 1,3-diphosphoglycerate. Now students, if it is diphosphoglycerate, that means what has happened? Phosphorylation would have happened, right? Phosphorylation would have happened. Yes, correct. Biphosphate, correct. Okay. Correct, yes, diphospho. Now, what is going to happen, students? We have to do phosphorylation, right? So, here in this arrow mark, I am going to write phosphorylation. If phosphorylation has to happen, that means I need ATP, I need phosphate, All of you agree to this? Yes or no? Yes or no? Is this much clear? Is this much clear? This is a little complicated process. That's why I'm going very slow. Okay, that's why I'm going very slow. Tell me if this much is clear. Right? Okay, that means I need 6 ATP, that will be formed from 6 ADP, and I need the phosphate, right? I need the phosphate to do this diphosphoglycerate. Can any of you tell me how many molecules of diphosphoglycerate should be formed? If six molecules of PGA has to be converted, will it be 6? Tell me quickly. Students, no spam, yaar, no spam. No spam. This is a very important topic. I don't want students who are really distracted to be here. If you're really distracted, please go and do not spam here, because I'm not able to see questions. I'm not able to see the answers. So no spam here. Those who are not interested, students, please. This is a very important topic. Yes. So how many molecules, can any of you tell how many diphosphoglycerate will I have to form? How many diphosphoglycerate will I have to form? Here if it is 6 molecules, here also it is going to be 6 molecules. 6 molecules. Agreed? Agreed? 6 molecules. Yes, 3 carbon but 6 molecules. Correct, Hanifa? That means I am going to get 6 into 3, which is your 18 carbon compound. Got it? Now, this is your first step. All of you write this down. This is your first step. Okay? That means 3 carbon dioxide was taken, 3 RuBP will accept it with the help of Rubisco, form 3 PGA which is 6 molecules. Okay? That is 3 PGA means 3 carbon PGA. Okay? 3 carbon compound. No, I'll erase this part, otherwise you will get confused. Okay, this is already, yes. So here 6 molecules, 6 into 3, I got an 18 carbon compound. Now here phosphorylation will happen. Phosphorylation, I need 6 phosphate groups that will be given up by the ATPs, and now I am going to get 1,3-diphosphoglycerate, which is 6 molecule. Again 6 into 3, that is your 18 carbon. So first step is clear. First step is clear. Right? Next step. Okay? Next step is, can I go to the next step? I want quick answers. 1 minute, students. Yes. Okay. Great. Now, next part. Reduction. Okay, reduction from diphosphoglycerate. Now reduction is going to happen. Who are we supposed to form? Who are we supposed to form? So I am going to write what I wrote here. I wrote 1,3-diphosphoglycerate will have to get converted to your phosphoglyceraldehyde, or I will write the short form. Okay, we have to get PGAL, that is your aldehyde, Now for this to happen, that means your reduction is going to happen. If reduction has to happen, you people tell me who should be doing the job here. If addition of hydrogen has to happen, NADPH has to do the job here. Agreed, all of you? NADPH has to do the job here. Yes, right? All of you are understanding? Yes? Quickly let me know. I can only see few of you understanding, others what happened, sleeping? Students, I am writing 6 here in blue color, that means 6 molecule, okay, 6 molecule. Here also if 6 molecules will form here, here also I am going to get 6 molecules, right? This is not the balancing, but this is 6 molecules. Now I have to have NADPH for reduction, that means NADP. Yes, got it? All of you? So this is your H. Okay, wait, I'll write it again. This is H. Okay, students, so who is going to do the job? Think about here, okay? Your diphosphoglycerate will be added with hydrogen with the help of NADP. Okay, NADPH. Now here we have 6 molecules. That means your NADP also has to be 6. Okay, NADPH also has to be 6 molecules. Okay, that will give us PGAL, that is your phosphoglyceraldehyde. Okay, glyceraldehyde. Now this glyceraldehyde, your 6-phosphoglyceraldehyde. Now let's see what your NCERT is saying. Reduction. These are a series of reactions that lead to the formation of glucose. The step involves utilization of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for reduction per carbon dioxide molecule fixed. The fixation of 6 molecules of carbon dioxide and 6 turns of cycles are required for the formation of 1 molecule of glucose. What are they trying to say? What I told you initially. 6 cycles have to happen for glucose to be formed, right? Now here we have taken 3 carbon dioxide. 3 carbon dioxide we have taken for us to understand easily. Now students, look here. Now next step is your regeneration. Now for regeneration to happen, what did we form? We formed PGAL. How many molecules of PGAL did we form? All of you tell me, how many molecules of PGAL did we form? Your phosphoglyceraldehyde. Quick, quick. Yes. Quickly answer me. How many molecules of phosphoglyceraldehyde did we form? Why is this so slow, students, to type one number? Fast. Type one number fast. How many molecules of PGAL did we form? Quick, quick. Okay, I'll give you option if it's too difficult. Is it 6 molecule or 3 molecule? Is it 6 molecule or 3 molecule of PGAL was formed? Give me if it's option A or option B. You have to be quick, yaar. What is this? 6 or 3 molecules of PGAL was formed. Yes? Yes. Quick, quick, quick. Yes. Okay, so 6 molecules was formed, right? 6 molecules were formed. So students, let's write here. 6 molecules were formed. 6 molecules. Out of the 6 molecules, 5 of them, that is 5 PGAL will go, and 1 PGAL, right? 1 PGAL will end up forming as glucose. Will end up forming as glucose. Okay, what am I trying to say? Out of the 6 PGAL, 5 PGAL will go for the regeneration of your Rubisco. Regeneration of your Rubisco. Because if regeneration of your that is your RUBP does not take place, not Rubisco, RUBP does not takes place, then C3 cycle will stop. C3 cycle will come to an end. So what is going to happen? Your 6 PGAL, 5 PGAL will go back to form the regeneration or more production of RUBP, and only 1 PGAL will form glucose. Now 1 PGAL has how many carbons? 1 PGAL has how many carbons? Now here we had 6 into 3, that was 18 carbons. That means 1 molecule will have 3 carbons, right? 3 carbons. Now is 3 carbons enough for glucose formation? No, right? No. So we need 2 carbons. That means this cycle which we did now with 3 carbon dioxide will repeat once again and give us 3 more carbons to form us a proper glucose, which is C6H12O6. C6H12O6. Okay. Now, why did we take 3 carbon dioxide? We took 3 carbon dioxide. Otherwise, if we take 1 carbon dioxide, we will have to calculate all of this in fractions. Because see, now that I have taken 3 carbon dioxide, it's easy for me to tell that 6 PGAL will be formed. Out of that, 5 will go here, 1 will go here. But if I would have taken 1 carbon dioxide, then I will have to tell this in fractions. So to avoid that confusion, we have done this calculation. Okay, now I'll show you an image and we will do a quick revision. Yes, so look here. Okay, so here we have taken 3 carbon dioxide molecules. So what is happening basically? Carbon dioxide from the outside is entering into your mesophyll cells inside your stroma, and then we are getting your sugar, right? Now here we have taken 3 carbon dioxide molecule with the help, okay, with the help of whom? With the help of RUBP and with the help of your enzyme Rubisco, we are going to form a 3 PGA, which is 6 molecules of PGA. Now that 6 molecules of PGA will has to get phosphorylated. That means phosphorylation will happen. Phosphorylation will happen. For phosphorylation to happen, I need inorganic phosphate, right? That I am going to get from whom? I am going to get from ATP. So I have taken 6 ATP because I have 6 molecules of PGA, right? Now this PGA, here I will get 6 molecules of phosphate. That will give me your diphosphor. Here students, one more step is there. That is your 1,3-diphosphoglycerate. That 1,3-diphosphoglycerate will undergo reduction. Now if reduction has to happen, I have 6 molecules, so I will write it here. I will add the step here so you will understand. Here what is that? 1,3-diphosphoglycerate. This 6 molecules are there. If I have to reduce this, okay, if I have to reduce, that means if I have to add hydrogen, that means I need 6 molecules of hydrogen. Who is going to give me 6 molecules of hydrogen? NADPH will give me 6 molecules of hydrogen. Now NADPH will do, NADPH will be added, it will give me 6 molecules of hydrogen. I will get the reduction will happen and I will get G3P or your PGAL, which I told you, okay, PGAL. Okay, that means 6 PGAL will be formed, out of which okay, out of which 5 your PGAL will get back to the cycle and do regeneration of your RUBP, and 1 PGAL, okay, 1 PGAL will end up forming your sugar, that is your glucose. 1 PGAL has 3 carbon. So that means this cycle has to go once again for me to get a 6 carbon to form the glucose. Okay. Now students, this will go here. Now here, if this PGAL has to become your RUBP, phosphorylation should happen here also. Okay. For that to happen, I am writing here phosphorylation to happen. Again, I need phosphate molecules. So for that phosphate molecule, who is going to help again? ATP is going to help. ATP is going to give me phosphate. Okay, 2 of the phosphate, 2 Pi or if inorganic phosphate will be lost, and the rest 3 will be used up here to form your RUBP. Are you all clear on this? Now we need to calculate the ATPs. Can I get a response, students? What is happening? Are you all clear on this? Yes or no? Students, I cannot see your response. Are you people? Yes, quickly let me know. If you understood so far, then only we can calculate the ATP and NADPH. Yes. Yes, tell me students. I am trying to see if you people are responding. Yes, okay. Is that clear? Is that clear so far? Yes, all of you? Okay, now I can see. Okay, students, can we calculate NADP and ATP? Now I can see the response. Yes, okay, great, okay. So students, here now we will calculate the ATP forms. Now look here, 6 ATPs, right? 6 ATPs here, 6 ATPs here, 3 ATPs, okay. So we will do the calculation here, 6 ATP plus 3 ATP. Now all of you tell me it is 9 ATPs. But in this cycle, did I form a glucose or should I go for the cycle once again? Tell me all of you. Shouldn't I go for the cycle once again to form glucose? Yes? Yes, quickly. Yes, right? Yes, once again, that means I have to do 9 ATP plus 9 ATP. That will give me how much? 18 ATPs. That means to form one molecule of glucose, I need to use 18 ATPs. I need to use 18 ATPs. Now let's calculate NADPH. NADPH, how many NADPH, students? Here I am taking 6 NADPH, right? 6 NADPH. Only here I am using? Only here I am using? Okay. Now here also I didn't form glucose. That means I have to double this. I have to double this. So I am going to write again NADPH. That will give me a total of 12 NADPH. Right? So totally how many ATPs and NADPH has to be formed to finish one glucose formation? 18 ATPs and 12 NADPH will form as one molecule of glucose. Are you clear on the cycle? Are you clear on the cycle? If all of you understood, quickly let me know in the chat box. Yes, very good. All of you? Is it easy now? Is it easy now? Great. Okay, students. Okay. Yes. Okay, I cannot see response as I am using. Okay. Now we will try to understand C4 cycle. See, understand students, C4 cycle is not this complicated, but there are some things that you need to understand. Okay. Once this much is clear, I will put this notes in the Telegram. Okay, I'll put this notes in the Telegram. So that's why I've made the markings. Now students, C4 cycle. Okay, we'll try to understand C4 cycle also very quickly. C4 cycle is mainly because the name comes because the first carbon compound is a 4 carbon compound, and this is mainly seen in plants like maize, okay, sugarcane, and many more plants. Okay, this kind of cycle is seen in maize, sugarcane, etc. Now I'll tell you what is the difference between this cycle. Students, understand, this cycle was first tried to be figured out by Hatch and Slack. They are the scientist name. Okay, so this is also called as Hatch and Slack cycle. So we can also call this like this. Okay, they were the people who tried to figure it out. We already understood a general idea that all the plants will not have C3 cycle. That means all the plants will have C3 cycle, but all the plants will not have PGA as the first stable compound. Okay, that is what we understood. Now C4 cycle is not seen in all the plants. Okay, C4 cycle is in specific like maize, sugarcane, etc. Now what is going to happen in your C4 cycle is, students, this C4 cycle, the adaptation, okay, the adaptation that C4 cycle takes is, adaptation is a dimorphic chloroplast. Dimorphic chloroplast. Okay. Now what is dimorphic? That means two types, right? Two types. Now dimorphic means granule, or it will have grana and agranule. Okay, granule and agranule. Now if I have to draw your granule and agranule, now your double membrane structure, right? This is your grana and this is your connections, right? Now it's the same thing I draw here, agranule. That means there is no granas, only stromal lamella will be there. Okay, stromal lamella will be there. Now what do you understand? So in granule there is grana that is present. Okay, here there will be no grana that will be present. This is a adaptation that is taken by your C4 cycle, taken by your C4 cycle. Now I told you one more thing, I am going to write here. I told you your rubisco will become oxygenase, or will do the job of oxygenation if the temperature is high, and if the oxygen level is high. If the oxygen level is high, correct? Now to avoid this, to avoid photorespiration, that means to utilize all the oxygen that is needed, to avoid photorespiration, your C4 cycle has a technique. That technique is this presence of grana and agranule. Now I will tell you what is it. Okay, students, now try to understand so much. Okay, now say I am drawing layer of cells. Okay, this is your mesophyll cells. Okay, and here we will have a vascular bundle. Okay, vascular bundle, and around this vascular bundle we will have, okay, we'll have a structure like this. Okay, students, I'm trying to explain you why this is called, also called as Kranz anatomy. Okay, that means your vascular bundle, around your vascular bundle, the structure will be looking like a wreath. Okay, that is why it is called a Kranz anatomy. Now look here, this is your mesophyll cells, this is your vascular bundle. I mean your vascular bundle will be lined with bundle sheath. Okay, I will label it now. I'll label it now. Say this is your mesophyll cells. Okay, this is the vascular bundle or your bundle sheath, and vascular bundle, this is your C4 cycle basically happens in the bundle sheath and the mesophyll cells. Is it visible to all of you? Visible to all of you? Yes, got it. Okay, students, so bundle sheath and your mesophyll cells. Okay, now understand that this is this whole C4 cycle can also be called as Kranz, or your C4 plants is having Kranz anatomy. Okay, what is Kranz anatomy? The presence of your bundle sheath in the form of a wreath around your vascular bundle. Now students, tell me, normally your C3 cycle will happen where? In the mesophyll cells, right? Here is where your C3 cycle will happen. And now in your C4 plants, okay, in your C4 plants, here C3 cycle will happen, right? Now this whole thing is actually facilitating for avoiding photorespiration. I'll tell you why. See, now here if the temperature is high, because the mesophyll cells will be exposed, right? Now here normally in normal plants, here is where C3 cycle will happen. But in your C4 plants, the C3 cycle will happen in the bundle sheath. C3 cycle will happen in the bundle sheath. Because of this, what is going to happen? Your rubisco will not get affected. How will rubisco not get affected? Look here in the mesophyll cells, the temperature is going to be high. So can rubisco get converted to an oxygenase? I told you, if temperature is high, rubisco will get converted or changed to an oxygenase in adding oxygen, right? Now tell me, here in mesophyll cells, if C3 cycle happens, will rubisco be affected or not? Tell me a yes or no. Quickly tell me yes or no. Yes, right? Yes, very good. Now in C4 cycle, that will not take place. Why? Because our C3 cycle is happening in the vascular sheath, and that vascular sheath is below, it's down, so it will have more warm temperature. So first way to avoid photorespiration is done. Second thing is what? Oxygen should be more. Oxygen should be more. Now tell me, in C3 cycle are we forming oxygen? In C3 cycle is oxygen getting released, or during our non-cyclic reaction? Non-cyclic reaction. Where is it happening? Where is it happening? In our non-cyclic reaction, correct? Non-cyclic reaction. Now that will also happen here. Then there will not be any extra oxygen for rubisco to get converted into oxygenase. Okay, is this much clear, students? Is this much clear to all of you? Okay, so C4 cycle have taken an adaptation where they will show two types of, or two stages of your cycle, that is in the mesophyll cells and in the bundle sheath. So whatever is forming in the mesophyll cells gets diffused into the bundle sheath and continue here to form your C3 cycle. So by this we will avoid rubisco to undergo photorespiration. Okay, to avoid photorespiration, your C4 cycle are capable of doing it. Now see, let's see the steps. Okay, let's see the step. I hope this much is clear to all of you. Yes, great. Okay, students, here I want to tell you one thing is, in your textbook that portion is not mentioned. Okay, that portion is not mentioned. So I thought I'll tell you people about it. Okay, so all of you are with me? All of you are with me? Yes? Yes. Okay. So students, in your textbook they have not mentioned about the enzyme formation. They have directly written HCO3. Okay, but understand that the carbon dioxide which is coming from outside, right, will enter your mesophyll cells first. So let's say I'll draw this mesophyll cells here, right? I'll draw the mesophyll cells here. Okay, now the carbon dioxide will enter and it will combine with the water that is present in the cell. Okay, that is present in the cell to form carboxylic acid. Okay, it will form the carboxylic acid. Okay, with the help of carbonic anhydrase. Okay, with the help of an enzyme called carbonic anhydrase. This is not mentioned in your textbook. So then this H2CO3 will again split to give H plus and HCO3. Okay, HCO3. This is what is mentioned in your textbook. That is why just understand this process. Carbon dioxide from outside will enter your mesophyll cells. So this is your mesophyll cells. And it will form the acid, and that will be done by carbonic anhydrase. That will again split into H plus and HCO3. This is what is mentioned in your textbook. Okay, now we will get into the step. Okay, look here. Plants that are adapted to dry tropical regions will have C4 pathway, and mentioned earlier, though these plants have C4 oxaloacetic acid as the first carbon dioxide fixation product, they also use C3 pathway. Very, very important. Most of them, what they will do, they will get confused that, oh, C4 plants will not do C3. No. C4 plants also will do C3, but they will do it in a better way. In a more expensive way. I'll tell you why I use the word expensive. Okay. Now, as the main biosynthetic pathway, then in what way are they different? Now, they are asking you people the question. NCERT is asking in what way are they different if they are doing C3 cycle also. In what way they are different? Can any of you tell me in what way they are different? In what way is your C4 plants different? I gave you the answer, so quickly tell me, let me know the answer. Yes. In what way your C4 plants are different if they are also doing C3 pathway? C3 pathway. Quick, more response. You can just tell one word. It's a one word answer. Okay, C3 in bundle sheath. Yeah, that is one way. Okay, what is the one thing that they are avoiding? Kranz anatomy, C4, C3 is occurring. Okay, correct. All of you are correct. I want more response. Tell me quickly, what is the difference? What is the main difference? Students, the difference is so much that C3 pathway is happening in the bundle sheath. Okay, it's happening in the bundle sheath, and it is also happening in a situation where there is no photorespiration. Right, I mentioned you now. Photorespiration is not happening. Because of that, the beneficial carbon dioxide, that means whatever oxygen was kept for use, no, will be still there. It will not be used up by this photorespiration process. That's all. Okay, now they have a special type of leaf anatomy. That is what I told you, Kranz anatomy. And see, look here, they show a response to high light intensity. They lack a process called photorespiration. This is the most important part. That means their productivity is more. Now let's see. Students, I'll explain you with the diagram. Okay, this is your so we will start from here. This is the first step. Okay, so the primary carbon dioxide acceptor. So what happened? I told you, carbon dioxide is coming, right? It is getting converted to HCO3. Okay, HCO3. Now that HCO3 will be accepted by an enzyme called PEP. Okay, PEP, that is your phosphophenol pyruvate, PEP, and it is present in the mesophyll cells. Now the enzyme is responsible for this fixation is your PEP carboxylase. So here, like how your RUBP was doing the job, here PEP will do the job with the help of an enzyme, called PEP carboxylase. Students, here one issue is not there. Can you tell me what is the issue? PEP carboxylase will only do fixation of carbon dioxide. That means this is only carboxylase. There is no oxygenase. So that tension is not at all there here. Okay. Now, it is important to register that the mesophyll cells lack Rubisco enzyme. Because of it, C3 cycle will not happen. Okay. Then the C4 acid formed is an oxaloacetic acid. So after this, what is getting formed? We are getting OAA. That is your oxaloacetic acetic acid, which is a 4-carbon compound, okay. Now it then forms a 4-carbon compound like malic acid. That means this oxaloacetic acid will again form malic acid, okay, malic acid. Students, in some cases this malic acid or aspartic acid also can be formed, okay. So, or aspartic acid. I'll show you with the diagram, do not worry. Aspartic acid is formed, which is a 4-carbon compound again. Now, this malic acid and aspartic acid will diffuse into the bundle sheath now. So, look here, which will be transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release 1 molecule of carbon dioxide and 3-carbon compound. Now, very important, very important. This you can all see, right? This malic acid, will get broken down into a 3-carbon compound, which is your pyruvate, and 1 carbon compound, which is your carbon dioxide. Now, this carbon dioxide will proceed to form the C3 cycle, or will enter into the C3 cycle, and this pyruvate can enter back, okay, it will enter back into the mesophyll cells, okay. I'll show you with the diagram so you'll understand more. Now, this 3-carbon molecule is transported back to the mesophyll, where it is converted to PAP again. Now, students, how many of you connect this thing with our Calvin cycle? Now, one thing is going back for regeneration of RUBP. Who is that? Who is that person who is going back for regeneration of RUBP? Quickly let me know. Yes, fast. Who is going back for regeneration of RUBP? Yes, very good, Trisha. PGAL, Sanvi, good job. PGAL, right? PGAL is going back to form RUBP. Here also, look here, your pyruvate is going back to form PEP. Again, otherwise this will get over, this process will get over, correct? Now, let's see with the diagram. Good job, students. PGAL, all of you have put Harini, Vishnu, good job. Now, look here. This is the diagram. So, what is happening? Atmospheric carbon dioxide is entering. It is going to combine. Okay, I'll write here. It is going to combine. Carbon dioxide is going to combine with water, form the acid, and that acid is getting broken down to H plus and HCO3. Okay, here that is what is written. Now, your HCO3 with the help of phosphophenol pyruvate and phosphophenol PEP carboxylase, it will get fixed, right? It will get fixed into a C4 acid. C4 acid is what? Your OAA. Okay, that OAA again will get converted to malic acid or aspartic acid, and it will diffuse into your bundle sheath. So, this is your mesophyll cells, this is your bundle sheath. This malic acid will again get into your bundle sheath, okay, and what will happen? This will break, right? That is your C4. Here I will write malic acid will get broken down into 2 ways, right? Into a 3-carbon compound, which is your pyruvate. Here they have written, students, and 1 carbon compound, which is your carbon dioxide. Now, this 3-carbon pyruvate, that is your 3-carbon compound. Okay, that is your pyruvate. Students, that pyruvate will go back. Okay, that pyruvate. See, you can see C3 acid. C3 acid means your pyruvate. Pyruvate will go back into this cycle for regeneration of your PEP, and this carbon dioxide which came out from here, this carbon dioxide will enter to do your Calvin cycle. Okay, this will enter to do your Calvin cycle. How many of you understood this? Tell me quickly. All of you got it? Yes or no? Yes? Understood. Okay, students, now we have to calculate the ATPs. Now, when this, that is your PEP formation, right? Now, when your pyruvate is going here back, no? When here it enters, here we need an energy formation. That is your ATP will become AMP. Now, what is AMP? Adenosine monophosphate. Now, what do I mean by adenosine monophosphate? Now, your A is there. It will be bonded with one phosphate, another phosphate and another phosphate. Now, if I break this bond, that means it becomes AP. That is what? Your adenosine monophosphate, right? Monophosphate. I'm only talking about the breaking of bond, okay? You can say, otherwise you can say 2 ATP molecules will break down to form 2 ADP. That means one molecule of ATP is breaking one phosphate bond. So, 2 molecules means 2 molecules of phosphate bond. Otherwise, here also same thing. See, 1 bond here, 2 bonds here. So, that means 2 phosphate bonds are broken down, correct? Now listen, 1 carbon dioxide. So, here 2 ATPs, correct? 1 carbon dioxide, I will erase this and we will do the calculation. I will write it here small, students, okay? So, ATP to AMP, okay? That means what? A molecule, phosphate, breaking down, right? Now, that means what? 1 carbon dioxide molecule has to be released. That means we are taking 2 ATPs. So, tell me, if I have to form glucose, I need 6 carbon dioxide molecules. That means how many ATPs should I have? How many ATPs should I have? Tell me quickly, students. How many ATPs is required if I need 6 carbon dioxide molecule? 6 carbon dioxide molecule. 1 carbon dioxide is requiring 2 ATPs here, okay, 2 ATPs. That means 6 carbon dioxide means how many ATPs? Yes, very good, very good. That means I will have to need, right? I will need 6 into 2. That means I need 12 ATPs. So, for just this reaction to happen, I need 12 ATPs. So, I need 12 ATPs for making my pyruvate regeneration to happen to PEP. Now, for Calvin cycle, how many ATPs do I need? For Calvin cycle, how many ATPs do I need? 18 ATPs, right? 18 ATPs. That means I need a total of 30 ATPs to synthesize one molecule, right? To synthesize one molecule. That means that is why I call this reaction as an expensive reaction. Your C4 cycle is an expensive reaction because we require around 30 ATPs to do the whole synthesis. That means here I need 18 ATP in the Calvin cycle. Here I need 12 ATP. 30 ATPs are required for me to complete my C4 cycle. So, is this an expensive reaction? Yes or no, students? Is it an expensive reaction? Yes, very good, Hanifa. Yes, it is an expensive reaction, but one thing benefit is that I am avoiding photorespiration completely. Photorespiration is avoided completely. Why? Because here there is no high temperature. Only here Rubisco will be there. Here Rubisco will not get affected by high temperature because definitely mesophyll cells are there. Bundle sheath is down. Okay, and here oxygen formation will not happen, right? Here oxygen will be formed, but here there is no accumulation of oxygen. So, more amount of oxygen is not there, right? So, by this way I can totally avoid, or the C4 plants can totally avoid photorespiration. Got it, students? Understood, all of you? Is C3 and C4 cycle understood? Quickly let me know in the chat box. Quickly like the session. So many of you are there, but the likes are so less on the session. I want each one of you to go like. Quickly. Yes. Is it crystal clear? Will you any time have a doubt? Will That's why I took it really slow. I went very slow in every step. Students, one important thing that you need to write in an MCQ question if they asked is, here in your mesophyll cells, Rubisco will not be present. Okay, Rubisco will not be present in this mesophyll cells. Normally, you know, if C3 cycle is happening, it will happen in your mesophyll cells where the chloroplast is there, correct? All of that we know. But then here it will not happen. Why? Because your mesophyll cells here, right here will have, this will be granule, okay, this will be, okay. So, understand this much, clear to all of you? Can I get quickly fire emoji in the chat box? Can I see quickly fire emoji in the chat box if all of you are clear, super clear? Yes, super clear on this. Yes, great. Yes, great. Okay, I want all of you to respond. Okay, I just don't want only 5-6 of them to respond, because I really took it very slow to make you understand. So all of you quickly react and let me know you understood, and like the session, okay. That's all. Only few of you understood, or are you people searching where's the emoji? Okay, okay, great. Yeah, some people are actually searching where the emojis. Okay, no problem. Students, if you have any doubts, feel free to comment in the, after this video, you can go and comment if you have any doubts. Okay, I will take all your doubts in the special class. And if you people want to join my special class, do join. The link is in the description box, where we have done really good, amazing content, molecular basis in one shot. We have done MCQs, we have done for most of the chapters. Yesterday we did excretion chapter MCQs. Quite complicated questions. So if you want any kind of like personal help, where I will be yesterday I even discussed, we had a last around 15-25 minutes of discussion about all of your doubts. So all that I can only do it on special class, not on YouTube. That's why I request all of you to go to the description box, click on the link, follow my profile, and you will get all the links, and there you can ask me any doubts that you have specifically, because here I cannot keep repeating, right? So you should come to the special class and I will clear your doubts. Okay, students? Yes. Yes. Oh, rapid fire. How can I forget? Yeah, sure. Rapid fire I'll do. Okay, rapid fire. Ready, all of you? Now I have to see here. So yeah, rapid fire. Yeah, there will be rapid fire, there will be MCQ. Every class of mine will have rapid fire, students. If I am taking it on special class, you will have rapid fire after every topic. Okay. Here it will be at the end of the topic. Okay, ready, students? Ready? If you are ready for rapid fire, you know what to do. All of you know what to do. First person's name will be called, okay? First person's name will be called. Yes. Tomorrow you will have a special class. Today you people have other classes, no? You have really important classes also. So tomorrow. Susan, tomorrow. Yes. Okay, ready. First question. Okay. For regeneration of PEP. Okay, for regeneration of PEP. What is the substance, or what is the organic compound that will enter back into the mesophyll cells? You can put the short form. Okay, for regeneration of PEP. Students, for regeneration of PEP, what is happening? All of you, what is? I am asking C4 cycle. I think Trisha has put it right. Students, pyruvate. I don't know why you people are putting RUBP. Pyruvate. PA. Pyruvate. Pyruvate is the one that enters back into the mesophyll cells for regeneration of your PEP. Yes or no? Most of you have put pyruvate. Sanvi has put pyruvate. Yes. Students, pyruvate is the answer. Okay, most of you got it right. Wrong. Listen to the question carefully. Listen to the question carefully. Okay. Next question. Next question. How many molecules of NADPH will be utilized for 3 carbon dioxide? That means if I am taking 3 carbon dioxide, how many NADPH will I need? How many NADPH will I need? Quick, it's a rapid fire. Think properly, think properly. 3 molecules of carbon dioxide, not 1 molecule. 3 molecules of carbon dioxide. How many molecules of NADPH will I need? Very good, Ganesh. Ganesh, very good. Trisha, very good. Nandini, Jyoti, very good. Sanvi, very good. 6 molecules of NADPH. 1 molecule of carbon dioxide will require 2 molecules of NADPH. That means 3 molecules of carbon dioxide will be double, 6 molecules, right? Okay, we have more. Yes, good job. Ganesh, good job. You answered first. Now, third question, students. Third question. How many ATPs are totally required for C4 cycle to happen? How many ATPs are required for C4 cycle to happen? C4 cycle. Total number of ATP I want. Nandini, very good. 30 ATP. Students, you have to be fast, okay? Rapid fire means you have to be fast. Very good. 30 ATP. 30 ATP. 18 ATP is for your cyclic reaction. 12 ATP is for the what happens in the mesophyll, right? Your C4 cycle. Totally we need 30 ATPs. That is why that cycle is called a expensive cycle. Okay, next question. Next question. Yes, carbon dioxide combines with H2O to form which acid and what is the enzyme? Okay, let's put it as 2 questions. First, tell me which is the acid that will be formed when carbon dioxide combines with water, and fifth question will be what is the enzyme that takes part in that. I had written the enzyme in the side. Very good, Nandini. Sanvi, good. No, Nandini, good. Sanvi formed the next product. Yes, good, Trisha. Carbonic anhydrase. Very, very good. So, the enzyme formed will be carbonic anhydrase. Okay, Nandini also put Jyoti, Ganesh, Hanifa, very good. Carbonic anhydrase, right? This will get broken down. This will get broken down to H plus and HCO3, which is given in your textbook. Yes, happy with the rapid fire? It is definitely, there is lot to remember, lot of ATPs to remember, what is happening, where it is happening, all this will be there. But students, definitely with one revision, you will be good to go. Okay? Yes, all of you are feeling a little confident, happy that you understood C3 and C4 cycle and it is not such a big thing. Yes or no? Quickly let me know, because I want to give you people breaks so that you can join the next class. Yes? Okay, great. Okay, students, thank you so much for joining, and do not forget to join our batches. Okay, Avengers 3.0 got launched. Okay, and those who missed the chance, please do join the exams mock series that is going to happen, right? That is already on process. So do not forget to join our crash courses if you want, or if you're thinking that can I write NEET in another 6 months. Definitely you can write NEET in 6 months. And if you are a dropper, especially you need some more, a proper guidance. So join us. We will help you with all the PYQs, MCQs, how to read your NCERT, how to make notes. All this will be dealt in our sessions. Okay. So do not forget to join. And the only thing that you have to do is use the code GOPIKA, GOPIKA. And it's not that such a big name, right? Thank you so much, students. Thank you so much. It was an amazing session, I hope. And I hope all of you will study this topic. Today itself, sit and read your NCERT, even if it's going to take 5 to 10 minutes. Read your NCERT, because this one, if it's not registered, you are going to keep forgetting it. Okay? Thank you so much. See you all. Take care. Have fun. Take rest. Okay. And join back for the next session.